Let's continue on. That was you? That was me. Well, the presentations beep. I mean, the recording beeps when they start off. Okay, so let me continue on with this. And yes, it is cold in here, and I know, and deal with it. Which is to say, always dress in layers when you come to this school, because there's no guarantee what the temperatures are going to be like. Next week you might come, all bundled up and they've turned the heating on. So be aware that that can happen. All right, so we were just finishing up talking about hormone receptors and closing out the idea of what hormones are. What I want to get into next is a few, oh shoot, I didn't want to do this yet. I said I was going to do the lab stuff, that's right. So we'll get back to that in a little bit. But so in this week's folder, I have a link here that says endocrine system micrographs, which takes you to a folder that's in a cloud source that I use that you can access it from anywhere. And I have a number of different micrographs that I've captured in one of the labs that we have here that I usually teach in, and hopefully in the future, the one that we're going to be using some this semester, we'll have this too. But there's a digital scope, so I can put images up on the screen and I can capture them and save them. And so I've done that as I went through a presentation about the endocrine system stuff in the past. So what I want to do is go through these. Well, let me just back out of this for a second. The figures are all labeled with what we're looking at. And then it says 4x or 10x or 40x, which is referring to the objective lens that's being used. It's not the actual total magnification because there are other lenses that this is going through. Part of the reason why I just put the objective lens number on there is because I'm not positive what the actual total magnification is through the digital camera on the microscope. The company that we bought the scope from was at a convention I went to last year, and so I asked them this question, and he said seemingly emphatically that it was the same as looking through a regular scope, but he couldn't really explain it too well, so I don't know if it's true. So whatever it is, this final magnification is not in fact 4x. It's just based on the microscopes we used, it's the idea that you're looking through a 4x objective plus whatever additional magnification comes from the system. So it's probably, somewhere in the 40 to 60 times magnification total, but I actually don't know. Now, I'll get to talking about what this image is in a little bit, but I want to set up the whole point of what I want to do here, which is to think about what we see through the microscope and how that will help us understand some of the anatomy and physiology of the endocrine system. Now, we're going to be looking at images that compare two tissues to each other. So like you can see here, there's a big crack down the middle of the tissue. And on one side, there's a darker half, and on the other side, there's a lighter half. And we're going to be looking at things where we can compare what these things look like side by side. I describe this as staining quality of the tissue, which really is just meaning how darkly it stains. So for you to understand what we're looking at and what the darkness of the stain means, I just want to review, or perhaps tell you for the first time, what actually happens when we stain tissue under the microscope. Okay. So the tissue we're looking at here has been collected from, obviously, some organ, and it's been sliced very, very thin. I don't know exactly how thin it's sliced, but for these kinds of sections, It's somewhere probably between 200 and 500 micrometers. Yeah, micrometers thick, okay, which is a fifth to a half of a millimeter. Okay. I'm betting it's closer to a quarter or so thick of a millimeter, but I actually have no idea exactly what it is. But very, very thin. Okay. If you were to just take tissue, cut that thin, and put it on a piece of glass, shine some light through it, look at it in the microscope, you wouldn't see anything. Because there's nothing inherently colored or opaque about tissue, especially when you take it apart at that level. When you can see color in tissue, when you're looking at an entire organ or something like that, most of what you're seeing is blood that's in that tissue. Because really, most tissue that you look at is going to be some shade of red, from pink to purple. You know, like, well, I guess you could say from white to purple. You know, tendons or the sclera of the eye, something like that's going to be white because it has very little blood in it. Okay, you're basically just looking at unadulterated tissue. Up to, say, the liver that has a lot of blood in it, so it's very dark red to purple looking in appearance. But when you're looking at tissue up close at very thin sections like this, it has no inherent color, unless you happen to be looking at tissue that has a pigment molecule in it. So if you were to take a section of skin and not stain it, but look at it in the microscope, you might be able to see some of the melanin that gives skin its pigment or something like that. But for most tissues, there's nothing inherently colored in the tissue. So to be able to see it, we actually have to stain it with a chemical dye. Now, the way I'm going to explain how this happens is actually a little bit oversimplified, not really how it works, but is essentially the idea and explains it pretty well. I'm actually kind of putting 2 or 3 steps in the staining process together so that you can see how it works. So first off, we've cut this really thin, and we've put it on the slide. The tissue needs to stick to the slide, so usually part of what's involved in that is fixing it, which is to soak it in something like a formaldehyde solution. Or there are other fixatives. Sometimes you can use ethanol, that'll fix things together, which is, it'll react with components in there and make them stick together and make it stick to the glass. And then you pass the tissue through a series of washes, depending on the exact stain. Sometimes it's 1, sometimes it's 3 or 4 washes that introduce the dye molecule to it and cause chemical reactions to happen between the dye and the tissue. And then you dehydrate the tissue because you don't want it to spoil, you don't want it to rot, so you dehydrate it so it's preserved. Which is usually passing it through a series of alcohol washes until you get all of the water out. Now, I'll get to the staining process in a second, which is important, but I want to mention the dehydration because that's important too, especially for what we're looking at. When you dehydrate something, when you pull water out of it, it's necessarily going to shrink. Okay? If you put banana slices in a dehydrator and to make banana chips, they're going to be smaller than they were. Okay? And that happens to tissue too. This big crack that we see here is not actually present in what this tissue is and naturally occurring in the body. Okay? There's not a gap between these two things. They're actually right up against each other. But once we've cut them thin, put them on the glass, when we dehydrate them, the tissues shrink. And the tissue just all shrinks up. And anywhere that there's a seam, a transition from one tissue to another, you're usually going to have tears in the tissue. And that's what that is. It's really just a tear, a crack in the tissue, right at the boundary between those two things, which is great for looking at it in a microscope because boundaries are often highlighted by that. Okay? You may or may not remember looking at a slide of skeletal muscle in AMP1, and the fascicles in blood and muscle fibers in muscle tissue, when they're dehydrated, shrink, and pull apart from each other. So you always get breaks in muscle tissue on endomysium and paramysium, which is great because then you can see the fascicles really clearly because they're going to sort of shrink together and you get separation between the fascicles. So that works pretty well. And here we see this break between two important parts of what we're looking at. Now back to the staining process. How this actually happens, like I said, is a little bit more complex than I'm describing, but the concept is essentially what I'm saying. The dye molecule is a charged particle, which means that it's going to react with other charged particles in the tissue. It's generally speaking, a positively charged particle, which means it's going to react best with what? It's positively charged. Negatively charged. The darker something stains, that means the more negative it is. The more negative the molecule is. Just the positively charged dye molecule just binds to it better and makes it darker, or more of the dye molecule binds to it, makes it darker. So that means the darkest thing that you see under the microscope in tissue is the most negative thing. Given that, and given what you know, having looked at tissue before because you were an AMP1, what's the most negative thing in any cell? The nucleus. And it's negative because the nucleus contains the most negatively charged molecule in your body. What is that? Molecule. DNA, right. What makes DNA very negative? No. It's, I mean, you could defend that position to some extent, but it's because of what's bonded in DNA. There's a specific part of DNA that's very negative.
Hydrogen.
A little bit bigger than that. And if it were hydrogen, hydrogen would be positive anyways. Like it is in the water molecule.
Is it because of nitrogen?
Sorry?
Is it because of the nitrogen bonds? Or is it...
No. It's not an atom. It's a group of atoms that we find over and over again in DNA. So what's DNA actually made up of? Okay. None of that's correct. Phospholipids are what make up cell membranes, and proteins are a completely different type of macromolecule. What are nucleic acids made up of?
Phosphate group, a sugar house.
Say that again.
The carbon house, the sugar house.
Yeah, the sugar thing,
Yeah.
which is ribose or deoxyribose, depending on which nucleic acid.
Yeah, the phosphate group.
A phosphate group.
Try to remember.
And another thing. What are those three things? The sugar, the phosphate group, and the other thing, which is called a base. What are those collectively called? It's a nucleotide, right? Right. Now, the actual answer that I'm getting at is one of the three parts that you just said. Okay. Those three things that make up a nucleotide. Which is the negative part?
Base.
Yeah.
Okay.
That's a good guess because if you're looking at things that are acids versus bases, oh, no, that is a bad guess. Acids are negative, bases are positive. But I thought you almost had me fooled. Yeah, but no. So it's not the base.
Phosphate.
It's phosphate. We're going to assume you said that out of knowledge, not out of a 50-50 guess. Phosphate is a phosphorus atom with 4 oxygen atoms bound to it. Phosphorus can make 5 bonds, which is not enough to completely bind to 4 oxygens. And so the oxygens have sort of incomplete bonds, and because of that, they're more negative than normal. They're not balanced parts of those molecules. So it makes the phosphate group rather negative. All right, So that thing with a phosphate group in it is called a nucleotide. It's one unit of the polymer that is DNA. Now, DNA is made up of two polymers. It's a double-stranded nucleic acid, double helix. It's got two strands that are wound around each other. What do we call one piece of DNA in your nucleus? What's a single DNA molecule referred to as?
The genome.
That would be all of them together in sort of a conceptual sense. What's one piece?
One gene.
Hmm?
Yes,
Gene is one region, but there are many genes together on one piece of DNA. So what contains a bunch of genes? You have 46 of them in any given cell. Chromosomes, right? They're numbered 1 through 22, and then there's the sex chromosomes, and those are paired. So 2 numbers 1, 2 numbers 2, 2 numbers 3, on down the line. The numbering is based on length. So chromosome number 1 is the longest piece of DNA. It has 291 million nucleotides in it. No, I should say that differently. 291 million base pairs in it. Okay. Remember, it's a double-stranded chain, and each chain is made out of nucleotides. 2 nucleotides bind to each other, connect to each other. That's a base pair, okay? AT or GC, base pairs. And there are 291 million of those in chromosome number 1. Okay. Each base pair is made out of 2 nucleotides. Each nucleotide has a negative phosphate group in it, which actually has a double negative charge. So it's -2, not just -1. So here's the math word problem that's going to haunt you at night. What's the charge of chromosome number 1?
Negative.
How negative?
Very.
Exactly how negative?
6 negative.
Okay. A phosphate group is -2. Okay. There are 2 phosphate groups in base pair.
-4.
So that's -4 for base pair. There's 291 million base pairs in that chromosome. And now that I'm saying it, I think that there's only one negative charge to a phosphate group. But that's beside the point. Really, really negative. Okay? It's somewhere around 380 some odd million negative charges. Needless to say, it's very, very negative. So the darkest things you see, which we can't actually see very well with this magnification, but let's say this little spot right there is a nucleus. Okay? We can see that very well because it has so much negative DNA that it picks up a lot of stain. We'll go to a higher power picture in a little while, and you'll see individual nuclei. You can pick that out a little better. But here, we'll pretend I have dots in nucleus. So nuclei are the most darkly staining thing you see in a cell, because DNA is the most negatively charged molecule in a cell. And you have a lot of DNA in the nucleus. What's the second most negatively charged molecule in a cell? DNA's first, what's second? RNA. Okay. Same reason. Made out of nucleotides. Nucleotides have phosphate groups in them. The reason it's not as negative as DNA is because RNA molecules aren't as long as DNA molecules. They're single-stranded, for one, so even if it was 291 nucleotides long, it'd only be half as negative as chromosome number 1. But actually, RNA molecules tend to be numbered in the thousands or hundreds long. Okay, it depends on the exact one, but basically that. Where do we see most RNA molecules in the cell? What was the second half what you said? Okay, yeah. In the nucleus is a fair thing to say. What I was really kind of getting at is that It's ribosomes, which is what makes rough ER rough. They're made in the nucleus, so they're going to be in the nucleus sometimes, but then they move from the nucleus into the cytoplasm where they attach to the ER. Now, not all ribosomes are attached to the ER. Some of them are just floating around free, and we call those free ribosomes. But whether they're free ribosomes or attached to the ER, they're in the cytoplasm. Okay. Now, there technically is a little bit in the nucleus, but that's masked by the very dark staining from the DNA. So after the nucleus being the darkest thing on the cell, then you can see the cytoplasm because of all the RNA in it. Okay. Now, what do ribosomes do? Yeah, they're there for translating proteins. So the more ribosomes that we see in a cell, it's capable of doing more of what? Right. We can pretty much say that the more ribosomes we see, then the more proteins that cell's capable of making. Or conversely, the darker the cytoplasm is, the more that cell's capable of making proteins. And the lighter the cytoplasm is, the less it makes proteins. Okay. So we're talking about staining quality, and it really comes down to the dark staining nuclei and the not so dark staining cytoplasm. That's what we're seeing. Now we're looking here at the hypophysis, which is another name for the pituitary gland. Now the pituitary gland is located just inferior to the hypothalamus. So if you wanted to call it by a French name, by a Greek name, it's inferior to something, so you'd want to call it hypo something. But you wouldn't call it the hypothalamus because that's just a weird construction. So it's called the hypophysis. And physis, which you probably can't really see up here, but that's the name, P-H-Y-S-I-S, means hypothalamus in that word. So it's below the hypothalamus. It means that in the sense that physis is the root of the word physiology. Hypothalamus is kind of the seat of a lot of physiology. And so the pituitary gland is just south of that. It's the hypophysis, hypophysis. When we talk about it as being the pituitary gland, we talk about the anterior pituitary and the posterior pituitary. When we call it the hypophysis, we talk about the adenohypophysis and the neurohypophysis. Now, I'm sure you know what one of those two is referring to. Which of those two versions of a hypophysis can you further define for me? Adenohypophysis or neurohypophysis?
Neuro.
Which means what?
It's got to do with the neurological functions.
Actually, it's more anatomical than that. It's made out of nervous tissue. Okay. So, of course, people are going to go there because you're used to the prefix neuro. Okay. And everybody avoids the other one because you probably don't really recognize the other prefix. But what does adeno mean? A-D-E-N-O. And you see that as part of a word. Can you think of other words that have that in them?
Adenine.
That would be true, but it doesn't help us answer the question. Yeah, that's kind of the same thing you said. I mean, he was talking about just, sorry?
Related to a gland.
Right. Adeno means related to the gland. I was hoping somebody might say adenoid, okay, which is a tonsil-like gland in your nasal or maybe oral cavity. But yeah, it's a gland. Adeno means glandular. Okay. So if the neurohypophysis is made of nervous tissue, then the adenohypophysis is made of glandular tissue. Okay. Now, so again, this is the 4x objective version of this picture. Oh, wrong direction. This is the 10x objective, and when we move to the 10x objective, we can start to actually make a nuclei. So I don't remember exactly what thing I pointed at, but now we can see little dark dots here. Those are nuclei. Okay. We can see it. more dark dots in here, which actually we want to kind of ignore that part of this picture because it complicates things. Over here, you can't really make out the darker dots because there's a lot of darkness surrounding them too, okay, which is cytoplasm. Okay. So this is the darker staining tissue, this is the lighter staining tissue. Okay. Which one's the adenohypophysis? Let's go with darker or lighter. Darker. Why do you say that?
Famously making, so I feel like making like hormones.
Right.
It's like glandular stuff. So.
Right. Glands are making and secreting hormones, and a lot of the hormones that we think about, and basically all of the hormones from the pituitary gland are protein hormones. Okay. So what you're saying would not really hold up if it was making lipid hormones, but it's not. So that's okay. So yeah, the darker stuff here is the adenohypophysis. Okay. And that would mean that the lighter stuff over here is the neurohypophysis. The adenohypophysis is a gland. It makes its own hormones and secretes them. The neurohypophysis is not a gland. It's really just an extension of the hypothalamus. There are a couple of nuclei in the hypothalamus that make hormones that are secreted into the bloodstream through the the neurohypophysis, which I slipped up there, is also known as the posterior pituitary. Okay. So posterior pituitary is nervous tissue, neurohypophysis. Anterior pituitary is glandular tissue, adenohypophysis. The hormones that are made and released, that are made in the hypothalamus and released from the posterior pituitary, very easy to remember. There's two of them. Okay? There's oxytocin and ADH, which stands for antidiuretic hormone. When we're talking about hormones, please use the abbreviations preferentially. Spelling's a lot easier, and you're less likely to get questions wrong that are autocorrected in Blackboard. Okay? And I'll tend to use the abbreviations. If I refer to something by its full name, that's probably because it doesn't have a common abbreviation. There are a couple of exceptions to that, but usually it is. So oxytocin, we don't really abbreviate. You will see some sources that abbreviate it, but I just prefer to use the whole name because it's actually quicker to say the whole name than to say the abbreviation for oxytocin. So, does anybody know what oxytocin does?
Helps with contractions during labor.
Right, so it's released from the hypothalamus through the posterior pituitary in response to stretch of the cervix, and it causes uterine muscles to contract. So it causes contractions during childbirth. It does a number of other things, but then that's its main thing. It's sometimes referred to as the love hormone because it's often associated with the generation of the emotional state that we consider love. And that might go with childbirth a little bit. Okay, a woman has to go through some pretty awful stuff, and at the end, like, oh, look at what we got. Oh, it's so cute. That's oxytocin. It's also, I think, released more in terms of milk letdown. It's not responsible for causing milk letdown, but it's present during that, which is going to increase bonding during breastfeeding and that kind of thing. There are a few other times that it's released, not necessarily in reproductive function all the time. I remember a while back, like when I was in college or something like that, it was rather common to refer to chocolate as having the same effects as oxytocin. I don't know if that's true. I don't know if that was something that some questionable study published back then, and it hasn't been supported since, or if it's just something that's fallen out of paper talking about. But sometimes I've heard that mentioned. And like I said, it's kind of an old association. So, but now, in the files that I have here, I just showed you the hypothesis at 4x and 10x objectives. I have a neurohypothesis specifically at 10x and 40x. And then I have the adeno hypothesis at 10x, 40x down here. I wish I had more control over how this website organized the files so that I could put them in some kind of order, but you just have to watch where they are. Okay, so we were just looking at this. If I go over next, this is just a 10x objective view, same magnification as the previous picture, but centered on just the neuro hypothesis. And if we go here, this is the neuro hypothesis now at high power using the 40x objective. And what you see here, the cells that we can see, the nuclei that we can see, don't really have cytoplasm surrounding them. Or at least it's definitely not visible. Okay, so there's not a whole lot of staining in the cytoplasm. What we're seeing, all of the space in between these cells, is really extracellular space, which is what nervous tissue looks like. When we look at nervous tissue, we usually don't look at this kind of nervous tissue. Okay, we're usually looking at nervous tissue where we see neurons, and there's actually not neurons here. These are probably all glia. Some of them are also blood vessel cells, but we don't really see any neurons here. So it's very lightly staining compared to the adeno hypothesis. So this is the 40x adeno hypothesis picture. And now we can see very nice dark nuclei here, and they're surrounded by pretty well stained cytoplasm because these cells are making a lot of proteins. They have a lot of ribosomes and rough ER and that sort of thing in them. Okay. Now, it's not terribly crucial, but it is in fact true. In most of these nuclei, you can see a little dark dot, usually somewhere in the middle. That's the nucleolus, which is in fact where the ribosomes are being made. So when I was asking where the ribosomes are or where the RNA is, the fact that you can answer that by saying in the nucleus is true because that's where they're made. And there's a fairly dark spot where that's taking place. But they move out into the cytoplasm and they give it its staining quality. Now this is fairly typical looking glandular tissue with one important aspect to it, which is the cells are kind of clustered together. So you see a group of cells here, and you see a group of cells here and here and here. In the anterior pituitary or adeno hypothesis, you see the cells clustered like that because there are a number of different hormones that are being made. And this cluster of cells all make the same hormone. And this cluster of cells all make a different hormone. And this makes a different. Now, in this field of view, we can see probably a couple dozen clusters of cells. There's only, I think, half a dozen hormones that are being made. So one cluster, like this one in the middle, might be making the same hormones as this cluster over here. They're just physically separated. But some of the clusters are making different things. Okay? And I think there's a table that addresses that, which I'll show you in a little while, but they're sort of clustered that way. That's the adeno hypothesis. Okay. And I just want to come back to the 4X. We can tell what's going on here just comparing the staining quality of the tissue here. One side's lighter, one side's darker. We're looking at glandular tissue versus nervous tissue, which illustrates a difference in the pituitary. The glandular half is the anterior pituitary. The nervous tissue half is the posterior pituitary. Okay. Now, I told you to ignore the stuff in the middle. Okay, here's a 10x version. That's a little bit darker than the nervous tissue, but not quite as dark as the adeno hypothesis. This little band right here, I'm not going to mention probably at all after what I have to say here. It's responsible for making its own hormone, which is also a protein hormone. So the cells there have some cytoplasm. They stain a little bit darker than the nervous tissue here. But the hormone that's made there is nothing that we're going to worry ourselves with. It's a weird little hormone. A previous book I used would say that hormone is only associated with particular disease states. And there was no evidence of it being present in measurable levels in normal individuals. Okay. Whether that's true or not, I don't know, because it's the only book that ever put it that way. But whatever it is, it's a very minor hormone, and we're just going to ignore it. And in fact, that little strip of tissue there isn't posterior pituitary. It's not even anterior pituitary. It's its own little sliver of tissue. And again, we don't care about it, so we'll just ignore it. But just sometimes students are a little concerned about the fact that this is obviously different appearing tissue, but it's not part of the anterior pituitary. So just ignore it. Remember, all of this lighter staining stuff is the posterior pituitary. Okay, so that's the pituitary gland. Now I want to show you the thyroid and parathyroid glands. Okay. Now, in this, hopefully you can kind of see that there's sort of three things going on here. This big thing back here is skeletal muscle tissue. Okay. It just happens to be in the slide because there's a muscle that runs right next to these glands. It gets captured there. But as far as we're concerned, we don't really care about that tissue. We only care about these other two tissues. So there's this big one here and this little one here, which are the thyroid and parathyroid glands. Now, the bigger one is the thyroid gland, which actually should be self-evident because the thyroid gland is huge, the parathyroid glands are small. And so if we take a section through these, the parathyroid gland necessarily has to be a little fleck of tissue next to a big thing that's the thyroid gland. Okay. So I've given it away. This is the thyroid gland. But what kind of tissue does that remind you of? Yeah, it looks a lot like adipose tissue. Why does it remind you of adipose tissue? What about the adipose tissue doesn't stain? The
actual fatty.
Yeah, the lipids, the fats in there. Being hydrophobic or nonpolar, they have no charge, and so they don't react with the dye molecule. Okay. And so they don't really stain. Now, they do kind of stain, but that's just because stains sort of get stuck in that part of the tissue. But it's not reacting with the stuff. So these big kind of grayish looking things here, that's the lipids. Okay. They're not unstained like the glass background here, because the stain molecules kind of get stuck in that part of the tissue, but it's not really staining the same way the really dark stuff that we see here is staining. Okay, so this is the 10x version of it. And we see the thyroid and parathyroid tissue a little bit closer. You can still kind of see that, lipid-filled aspect of it that reminds you of adipose tissue. But as you get to look in closer, you'll notice something that really is not like adipose tissue, which is there are a lot of little dots or nuclei surrounding the lipid deposits here. With adipose tissue, that lipid deposit is a vacuole inside of a cell. Okay? So there can only be one nucleus around that lipid deposit because cells only have one nucleus. But we see a bunch of nuclei surrounding these things, which is even more obvious when we go to the 40x objective view. Okay, so there's this big lipid space here, and then these are nuclei of cells that are surrounding it. Okay. So it's, from afar, it really does look like adipose tissue, but when you get up close, it's very different because of the relationship of the cells to the lipid sections. This big lipid deposit and the cells that are surrounding it collectively are called a thyroid follicle. The cells are called follicular cells because they're part of the follicle. And they're responsible for making and secreting the thyroid hormones T3 and T4. Now, T3 and T4 are derived from an amino acid, but the polar aspects of that amino acid, the amino group and the carboxyl group that makes it an acid, and a few other things are removed, and all that's left over is a hydrocarbon ring. Okay, and those hydrocarbon rings are the basis of T3 and T4. Now, the full name for T3 is triiodothyronine or something close to that. I might be getting it a little bit wrong. But what's important is the triiodo part. Okay. What does tri mean? 3. So that's why it's called T3, because it's tri. What does iodo refer to? Might be a little bit of a stretch, but think about putting the letters INE on the end of that. Iodine, right? Thyroid hormones, T3 and T4, have iodine associated with them. And triiodothyronine has 3 iodine atoms integrated into the molecule. I can't remember now if T4 has 4 of them or not, but that's not terribly important. Iodine is a necessary part of the thyroid gland's function to make those hormones. If it doesn't have iodine, it can't make those hormones, and it causes thyroid problems. Does anybody know what the very obvious result of low iodine levels is on the thyroid gland? What? Not weight gain, but an increase in tissue. That's very, very obvious. There's a picture of it in the book, actually. It's called a goiter. So here's a picture of a goiter. And because of the lack of iodine, the thyroid gland starts sort of growing out of control and causes problems. It's a very striking problem and seems horrible, but can be fixed very easily by increasing iodine intake in the diet. Now, goiters are not very common in a developed country like the United States because iodine is a very common part of our diets. How do we get iodine in our diet? Yeah, it's added to salt. Now, salt is sodium chloride. There's no iodine in it. But when you go buy the stuff with the little girl in the umbrella, it's trademarked, I'm not naming it, but it's iodized salt, okay? Because they've added iodine to it so that it's in your diet. And in underdeveloped countries where you do not go to the store to buy a blue package with a little girl and an umbrella on it, you get your salt from more natural sources. And naturally, salt is not iodized. I saw a picture or a video, I don't remember exactly which now. If it was a picture, I just animated it in my head, or I actually saw a video of this, of some people in coastal parts of India getting salt. And what they do is they put on a whole bunch of clothes, and they walk out into the sea or ocean, whatever you want to call it. And then they'd come out of the sea and they take off all their clothes and lay them out on rocks to dry. What would happen at the end of that? Yeah, the clothes would be crusted with salt. Okay, so they get sea salt very easily that way. It's just that sea salt's not going to be iodized because it's a natural source. So they aren't going to get iodine in their diet easily the same way we can. Now, iodine's available in other foods, I'm sure. I don't know offhand what. If it wasn't, then there's no way that the thyroid gland could have evolved to require iodine. It had to be a source in our diet for it. I just don't know what it is. You know, it might be spinach or something like that. So, and I say spinach because spinach is something that's not uncommon in Indian food. So, so there's probably a good dietary source, but you know, Americans don't eat their vegetables, so they have to get iodized salt, whatever. So a goiter is just when these cells that make T3 and T4 don't have access to iodine, and then that creates problems, and the thyroid gland basically starts enlarging. And just a simple dietary source of iodine fixes that problem. Now, not all of the cells here are actually surrounding a lipid deposit. So there are some cells like this, perhaps, that are not part of a follicle. And we call those parafollicular cells. They also make hormones, but they don't make lipid-based hormones or lipid-soluble hormones. Okay. T3 and T4 need to be made in the follicles because this big lipid deposit is necessary for an environment for the chemistry to take place. But these cells here don't have direct access to lipids because they don't need it. They make a different hormone, which is a protein. So they can make it like any cell makes proteins. That hormone, which I mentioned earlier, is calcitonin. Okay? And calcitonin is just a plain old protein hormone. And it's made by the parafollicular cells of the thyroid gland. Now, in this 4X objective picture, I can't actually guarantee that these are parafollicular cells. Over here, it might be a little bit more obvious that this is probably the edge of a follicle that, it's like if you took a orange, for instance, and you cut right through the middle, then you'd see the orange peel on the outside, obviously, like what's surrounding the thing. But if you cut off the edge of the orange, you're going to be cutting through the peel. And so this might be cutting through the cells that are surrounding a follicle. We just can't quite see it. So I don't know necessarily that those are parafollicular cells or not, based on the information we have. But if I go back to the 10X version, I feel fairly confident, especially that over here, a lot of these cells are parafollicular. Or this little cluster here is probably parafollicular. I'm not exactly sure which area that previous picture zoomed in on, but I think those are pretty obviously parafollicular cells. The reason I say that is if I could isolate just that little square of tissue right there, and then go over the parathyroid gland and highlight the same size square of tissue, you wouldn't be able to tell them apart, okay? This cluster of cells here is about the same as any comparable sized area of cells in this gland. Okay? What makes the thyroid gland looks lighter overall is all of the follicles and their lipid deposits. But the parafollicular cells essentially are the same as any other gland. They're making protein hormones, and there's nothing special about them. But I don't have a picture looking at just that little area up close like that. Okay. But you could kind of see that this gland here and this gland here, parafollicular cells of this gland here, are probably equivalent cells, staining quality-wise. And they are basically the same. Now, the parathyroid gland is responsible for making only one hormone, which is called parathyroid hormone, or more easily PTH. Okay, that's the name of the hormone. Parathyroid is the name of the gland or the hormone. It's not appropriate to use it for one or the other by itself. If you're talking about the hormone, it's parathyroid hormone, which is abbreviated PTH. Okay? All of the cells here make that. Now, I should have a 40X version of that too. Yeah, there we go. So here's that a little bit closer up. And unlike the adenohypophysis, all of these cells seem to be kind of a uniform appearance. They're not clustered like in the pituitary gland because they're all doing the exact same thing. There's no reason to cluster them into separate groups that are responsible for doing separate things. These are all doing exactly the same thing. And the cells might be a little bit smaller here, but that's not important. So that's the thyroid and parathyroid glands. Okay. And again, over on the side here in the 4X, that's a muscle because there's a muscle that's right next to those glands. And so it's in this section. But if we look at the two glands alone, we can see the darker parathyroid hormone, which is densely packed cells, and the lighter thyroid hormone, which is lighter because of all the lipid deposits in it. Okay. But really, if you get close up, the strict glandular cells of the thyroid gland, the paraglandular regions, and the parathyroid glands actually look pretty much identical. Now, be careful. The prefix para is used in two instances which can get confusing. Parathyroid gland makes parathyroid hormone, PTH, to help control calcium. Parafollicular cells of the thyroid gland make calcitonin, which is also responsible for controlling calcium. Now, those two hormones, calcitonin and PTH, play two sides of the game. One increases calcium levels and the other decreases calcium levels. Okay, so if your blood calcium levels are high, PTH lowers them. If your blood calcium levels are low, calcitonin raises them. Where do you keep most of the calcium in your body? In your bones. So that's what their effect is. Calcitonin moves calcium from the blood to the bones. PTH takes calcium out of the bones and gets it back into the blood. Okay. There's a little bit more to what they do, but that's essentially what they're about. So after the thyroid, parathyroid group, I want to go to the pancreas. Now, here we're looking at just one gland, but within the gland, there are two types of tissue. There's a darker and a lighter staining type. Now, those tissues are endocrine tissue and exocrine tissue. What does exocrine mean?
Like outside of the body.
To secrete outside of the body. Crine means to secrete, so exo means out of. However, it might seem confusing because when you think of what's outside of your body, you probably are thinking of things outside of your skin. Okay? But exocrine functions include secretions that to your conscious perception of your body seems like inside, but as far as say your immune system or the endocrine system is concerned, is outside your body. Okay. That includes the lumen of any mucus-lined tract. So the respiratory tract, the digestive tract, the urinary tract, or the reproductive tract, the lumen of those tracts is effectively outside your body. That's largely an immunological definition, but it applies here too. So the exocrine function of the pancreas is to secrete digestive enzymes into the lumen of the small intestine, which is the digestive tract. And then the endocrine function is to secrete insulin and another hormone that regulates blood glucose levels. So I'm going to zoom in on this little area right here where we see a little lighter region surrounded by a darker region. Those lighter patches that we see in the pancreas are called pancreatic islets. Islet, it means little island. So it's like little islands floating in a darker sea around them. And I happen, as I change to 10x, 40x, I'm zooming in on that one right there. Okay, so here's a 10x. Here's that islet. And now we can see a little bit more detail. There are nuclei visible in the islet, and there are nuclei visible kind of in the darker area too. The nuclei in the darker area are kind of visible because it's such a dark area. At this level, we can't really see the nuclei, but they're there. Kind of like with the anterior pituitary gland. Okay. So let's move to a higher magnification. And so here's that same islet. We can see the nuclei here still pretty well. Now we can make out the nuclei in the darker staining tissue. And the reason we couldn't really see them before is that they're surrounded by very dark cytoplasm. Okay. There's some lighter cytoplasm and darker cytoplasm and the nuclei sort of in the darker cytoplasm area. Okay. Whereas the lighter stuff that we see here in the islet, you can pretty well make out the nuclei and relatively light cytoplasm there. Now, the lighter areas are in fact the endocrine tissue. Okay? The darker areas are the exocrine tissue. And that just has to do with how much protein either is making. Okay? The amount of protein necessary for for blood glucose regulation, meaning the amount of insulin or other hormones involved, is pretty minor. It just has to be enough to activate receptors. Whereas the amount of protein necessary in digestive juices is immense. You've eaten a pizza, okay? And there are a bunch of proteins involved in what was on your pizza. And so you have to make a bunch of enzymes to break down those proteins. And there's a bunch of carbs in that pizza crust. You have to make enzymes to break down all the carbs in the crust. And there's a bunch of lipids in the cheese on the pizza. and you have to break down those lipids with enzymes. So you're making scads and scads of enzymes to break down the food that you eat. And so these darker areas are just making lots and lots of enzymes to be released into the small intestine. So the darker part is the exocrine digestive function part, and the lighter part is the endocrine blood glucose regulating part. Now, there are two types of cells in the islet. We cannot actually tell them apart as we're looking at it now. To be able to actually identify the two types of cells, we'd have to use a different type of stain, which we just don't use. So I can't say that is one type and that is another type of cell. Okay. They are referred to as alpha cells and beta cells. Okay. The alpha cells tend to be at the edge of the islet, and the beta cells tend to be towards the middle. Okay. So if I point at this cell right here, since it's in the middle of the islet, it's safe to say that it's probably a beta cell. Or if I point at this cell here at the edge of the islet, it's safe to say that's an alpha cell. It's not absolutely conclusive. I might be pointing at a beta cell and calling it an alpha cell or vice versa. But the majority of the time, it's correct to say alpha on the edge, beta in the center. Now, let me start with beta cells. Beta cells make insulin. Okay. Alpha cells make the counterpart to insulin, which is called glucagon. Just like calcitonin and PTH balance calcium levels, insulin and glucagon balance blood glucose levels. Okay. When blood glucose levels are high, you release insulin to lower blood glucose levels. When blood glucose levels are low, you release glucagon to raise blood glucose levels back to the normal range. Okay. And I'm going to show you a little bit more about that in a little while. But what we can see in the tissue is the lighter staining part of this tissue is the part that's responsible for releasing endocrine hormones and controlling that. And the darker staining stuff is glandular tissue releasing enzymes for digestion. So then we come to the adrenal cortex, or adrenal gland, which is the last part of what I want to show you here. Now, what I've taken as a picture here is kind of half of the adrenal gland. The middle of the adrenal gland is the adrenal medulla. And then from the edge of the medulla to the edge of the gland is referred to as the adrenal cortex. The adrenal medulla is really part of the sympathetic system, sympathetic division of the autonomic nervous system. That's where epinephrine, also known as adrenaline, is being released as part of the fight or flight response. The cortex is strictly endocrine function, and it makes a number of different steroid hormones called corticoids or cortical steroids to be released. You can see from the edge of the gland here to the edge of the medulla, that whole area there, there's some variation in staining quality. The outer part is a fairly densely staining dark area with a very distinct line. And then from that distinct line, we have a fairly light staining area that blends into a darker staining area that ends at the distinct line, which is the border with the medulla. Okay. Oops, sorry. I should have gone the other direction. Okay, so this is the picture I was just looking at. If I go the other direction, this is a 10x objective view showing the border between the adrenal medulla, where adrenaline comes from, and the adrenal cortex out here. Now, it's not showing you the entire cortex, just the dark fading into light part before, and then here's just that same picture moved over. And then the light part ends right here at this edge where we have this final part at the edge. Now, here, this picture is to show all of the adrenal cortex. I need to go to, the book real quick, or I'm going to say this incorrectly. So this is a micrograph of the adrenal cortex, just like what I'm showing you there. And these brackets point out the different regions. So this micrograph here, it's just the same as this micrograph here, except that we can see a little bit of the medulla in the book version. And if there's any medulla, it's right here at the edge of this, but it's practically not in the picture at all. Okay. So adjacent to the adrenal medulla, the deepest part of the cortex is called the zona reticularis. And that's the darker part that fades into the lighter part. And that lighter part is called the zona fasciculata. And then the outer, very distinct edge part is called the zona glomerulosa. Okay. So in here, this part out here is the zona glomerulosa. And then where it starts to be the light staining area, down here until it fades into the dark, this is all the zona fasciculata. See, I got to have this in front of me or I'll say I'm wrong. And then the dark staining part here to the edge, that's the zona reticularis. Okay, good, I got that right. Okay. So just to orient you to this picture, but so that I have it in front of me and I say it correctly, let me go back to this picture. Okay. So like I said, the adrenal medulla is responsible for releasing epinephrine, which is part of the fight or flight response, also called adrenaline. It also releases norepinephrine, also known as noradrenaline, but that's minor. Most of what it releases is epinephrine. Okay. Norepinephrine we think of more as the neurotransmitter released from postganglionic sympathetic fibers. Although, yes, it is true that it's a little bit's released here, but that's minor. Okay. The zona glomerulosa releases what are called mineralocorticoids or mineralocorticoids. Most times when I see this word, there's an A between the L and the C, so it's mineralocorticoids. But the spelling's not that important. Okay. The mineral part of the word refers to the fact that they regulate mineral balance. Okay. Corticoid means that it's a steroid hormone from the adrenal cortex. And an example of that is aldosterone, which is responsible for regulating sodium levels, sodium being a mineral. Then when we go into the lighter staining zona fasciculata, that's responsible for making glucocorticoids, which regulate glucose metabolism. Now, it's not the same as what insulin does, which is regulating glucose levels in the blood. This is telling cells what to do with the glucose, okay, which insulin plays a little part in that too, but this is under special conditions these are used. Here's a few examples. You've probably heard of cortisol, okay, which is actually referred to as often as stress hormone. When you're stressed, you release cortisol, which is going to stimulate glucose metabolism, which means you're going to start having cravings for sweet things, something like that. So when you get stressed, you got to have a cupcake or whatever, blame it on the cortisol. Okay. Don't use that as an excuse to eat all the cupcakes you want, but that's part of what's behind it. The darker part here is the zona reticularis, which is responsible for releasing different sources will use different names. In keeping with what I've already told you, mineralocorticoids, glucocorticoids, we can call those gonatocorticoids. Gonado referring to the reproductive glands. Other sources will just call them sex hormones, and in the book here they refer to them as androgens. Okay. I don't tend to say androgens because I'd like to use a more general term, because androgens are testosterone and related molecules. Okay. Sex hormones or gonatocorticoids will allow for including estrogens or progesterone, which I think are also released from here, but I might be mistaken. So I'm not comfortable saying androgens exclusively because I want some room for there to be other sex hormones being possibly related there, but probably most of it is androgens. Now, androgen means to generate masculinity. Andro is a prefix or a root that means man. Okay. Estrogen means to generate feminine characteristics. Okay. Androgen and estrogen are equivalent words. Estrogen's not the name of a hormone, it's the name of a group of hormones. Just like androgen's not a hormone, it's a group of hormones. But a good example of an androgen is testosterone. A good example of an estrogen, which I think I mentioned earlier, is estradiol. Okay. And again, we're going to talk about this stuff more in the reproductive system, but I just want to lay a little groundwork for when we get there that estrogen is a group of hormones, not a specific hormone. So those three layers there, the glomerulosa, the fasciculata, and the reticularis, are both responsible for making different types of corticoids. Mineralocorticoids, glucocorticoids, gonadoccorticoids. Now, when we look at the anatomy here, there's a very distinct line separating the glomerulosa from the fasciculata. And there's a very distinct line that we can't really see in this picture, but we can in this picture, separating the medulla from the reticularis.
Yeah.
Okay, good. Oops, wrong direction. But there's not a very distinct line between the fasciculata and reticularis. Okay. When there's a weak anatomical sign, structure, indication, it usually means that there's a weak physiological aspect to it also. And the physiology of these cells here is that they release particular hormones. And it turns out that things aren't quite as distinct as are said here. Okay? Mineralocorticoids are what are released from the glomerulosa. Glucocorticoids are released from the fasciculata and the reticularis. Okay? So all of the area except for the glomerulosa releases glucocorticoids. But the reticularis adds to the glucocorticoids the fact that it releases androgens. Okay. Now, the reason why those actually kind of go together is that androgens are usually anabolic steroids. Okay, we talked about that earlier, building muscle mass, abusing that in sports as a performance-enhancing drug. If you're going to increase muscle mass, you need energy for that muscle mass to grow. So the area that releases androgens is also going to release something to stimulate glucose metabolism to provide energy for the anabolic processes that androgens are going to be responsible for. So they do act, physiologically, they do have to go together. And there's not a distinct border between those two layers because actually there's not a distinct physiological separation between them. The darker cells are responsible for releasing androgens, but all of the cells release glucocorticoids, which is not really highlighted and is usually overlooked, but it is true because the effects of androgens will also require energy in the form of glucose metabolism. So you need both of those functions tied together. So that's the adrenal cortex. Now, if we go back to the list here, you'll see that, oh, never mind. Oh, no. Yeah, I can say this. You'll see that for the adrenal gland, I don't have any 4DX pictures. Okay. Looking closely at the cells isn't terribly important. What's important about the adrenal gland is seeing the color or staining quality changes, dark to light to dark, back to light, so that you can see the different regions. We've already looked at the cells up close, 4DX and the other things, so you can see what glandular cells look like. So I don't have a 4DX for the adrenal gland because I really want you to concentrate on sort of the overall picture. And even this little thumbnail here, you can see the dark band of the glomerulosa, the lighter area of the fasciculata, and the darker part that's the reticularis. And you can just see a hint of the light edge of the medulla there. So even at this very low power picture thumbnail here, you can make out those staining qualities of it. So I don't have a high power picture because that's not what I want you to concentrate with this. It's really just about the staining quality. and relate that to the particular hormones that are released from those different areas. So that runs through the different tissues and gives you an idea of how staining quality tells you something about the function of those tissues. Staining quality is really an anatomical feature, how the cells look under the microscope, but it corresponds to what the cells do to some degree also, whether it's glandular versus nervous tissue in that some make hormones and some don't, or lipid-rich versus straight glandular tissue because of the types of hormones that are made, or dark versus light glandular tissue based on how much the tissue is making, that being the digestive enzymes versus the hormones in the pancreatic islets, or the variation in staining quality that corresponds to the different types of hormones made in different regions of a gland. Those staining qualities can tell you something about what they do. So for the lab material of this section, that's what it's about, looking at those micrographs, which you have access to through Blackboard online. Okay. If you really want to look at these under the microscope yourself, you're welcome to. We won't have time to do it in class, but you're always welcome to use room 511 down the hall, which hopefully you used while you were in A&P 1, but if you weren't. Room 511 down the hall has basically all of the stuff that's in an anatomy lab, and you can look at it. And so far, all we've really done anatomy lab-wise is these tissues. They are in the yellow slide box, and there's a yellow slide box down there, maybe more than one. These 4 slides that we've looked at, sure, we've looked at lots of pictures, but really only 4 slides. It's slides number 11, 12, 13, and 14 in that box, in the order that we looked at them. 11 is the hypophysis, or pituitary gland. 12 is the thyroid, parathyroid tissues. 13 is the pancreas, and 14 is the adrenal gland. So if you want to look at those slides, that's where they are in the boxes. We might get microscopes out later on for a different unit. So if you'd like to, while you have those out, look at these slides in lab, you're welcome to. Or you can, again, use room 511 whenever you want to. We keep that room locked, but if you ask me or one of my colleagues to open it, we'll let you in. We just keep it locked so that non-A&P students don't walk away with things. And A&P students, we don't want walking away from with things either, but we trust you because you're A&P students. Anyways, so that's the lab stuff. Let me go back to the presentation. I want to touch on a few things. I only have 20 minutes to touch on a few things, so I'm going to touch very quickly because I'm going to hopefully have a lot of this stuff available for you to watch outside of class soon enough. So hypothalamic pituitary relationships, here's just the anatomy. The hypothalamus is this area here, and then the pituitary is just below it. There's an anterior and a posterior portion to it. The posterior portion is where axons from a couple of nuclei in the hypothalamus, those axons end and release hormones into the bloodstream. Now, these are neurons, but they're referred to as neurosecretory cells to highlight the fact that they are neurons that don't make synapses. Okay? Where they would make a synapse with something, instead, the axon terminal ends on a blood vessel and releases the hormone into the bloodstream. And one of those nuclei makes oxytocin, and the other one makes ADH, or antidiuretic hormone. We talked about what oxytocin does. It's the one related to reproductive function. Antidiuretic hormone helps to regulate fluid levels. Diuresis, it means to pass water, okay, i.e. urinate. Antidiuresis would be the opposite of urination, or retaining water. So antidiuretic hormone is a hormone that causes you to retain water from your kidneys and not release it in your urine. ADH is most active overnight, when you're asleep and you're not drinking, so you retain water to keep your body with the proper fluid levels at night. Okay. Oh, whoops. Oh, I see what it did. So this is the anterior pituitary. There are hormones released from neurosecretory cells of the hypothalamus onto blood vessels that are around the infundibulum, or the pituitary stalk, the connection to the pituitary. Those blood vessels go down and surround the anterior pituitary, and the hormones that are released here just travel that short distance and affect the cells in here. The hypothalamus is releasing hormones that regulate the anterior pituitary, and then the anterior pituitary will release hormones that affect other parts of the body. Okay, and I'll have an example of that in a little while. Now, I went past this real quick. This is a table showing the different anterior pituitary hormones. Okay, I'm going to talk a little bit about growth hormone, which thankfully they're not calling human growth hormone here. It is just growth hormone, abbreviated GH. I'll tend to refer to it as growth hormone because it doesn't save you any time saying GH as opposed to growth hormone. I won't talk about prolactin, but that's the one that actually stimulates milk production. I won't talk about TSH, but obviously from its name, it stimulates the thyroid gland. I won't talk about adrenocorticotropic hormone, or ACTH, but it stimulates the adrenal cortex to release the hormones that we just talked about. And then I won't talk about FSH or LH right now, but they're very important for reproductive function. So we'll come back to them in the reproductive system, where they stimulate gamete production, which is egg or sperm production. That's what follicle stimulating hormone does. And they stimulate the production of, it says androgen here, and there technically is a good reason for it saying that, but the sex hormones, okay? So testes release testosterone, the ovaries release the estrogens and progesterone. But the story is a little bit more complicated than that. But that's what they will do. So we will talk about these, but not until the reproductive system. Okay. Then I got the sequence here out of order, and I'll fix it later on. But the two more that we can see here, antidiuretic hormone and oxytocin, I just mentioned, are coming from the posterior pituitary. They're made by the hypothalamus, but they're released at the posterior pituitary. Oops. All of these that I just went through here, the anterior, they're made in the anterior pituitary and released there. Posterior pituitary is not a gland. It doesn't make hormones. It just releases those two that come from the hypothalamus. I'm not going to talk about this picture, but I want to point out something that you should pay attention to, that there is negative feedback systems in how the hypothalamus and pituitary regulate other things. So this is sort of an explanation of that from the book, which I'm not going to talk about because I'm going to hopefully get to talk about it in a specific example in a second. So I do want to quickly mention some things about the pancreas. It is located posterior to the stomach. The stomach's not in the picture here because it would block our view of the pancreas. It's also kind of right in the crook of the beginning of the small intestine. Okay? And in fact, the pancreatic duct connects to the small intestine where it releases those digestive enzymes that I talked about. But scattered around within the pancreas are these little islets, which are responsible for making insulin and glucagon. I seem to remember reading somewhere that the islets make up about 10% of the structure and function of the pancreas, and the rest of it's like 90%. I don't remember where I read that. From this picture, it's believable. But usually when something is 90-10, that's a very gross oversimplification of something. So I don't tend to trust 90-10 splits too often, but it could be true. I just don't remember where I heard that. So I haven't been able to verify. But there's very little in endocrine tissue, and there's a lot of exocrine tissue in the pancreas. This picture is a neat little figure 8 sequence showing how insulin and glucagon do their jobs. Now, lots of textbooks will have something like this. I like how this textbook is using a little finger prick blood testing machine to talk about the values that we're looking at here. So normally your blood glucose level should be in the 70 to 100 milligrams per deciliter range. And so 90 would be an appropriate level there. When the levels are low, when you're hypoglycemic, and so here it's saying 50 milligrams per deciliter, which would count as that, then glucagon is released from the islets in the pancreas, which targets mainly the liver. Okay. Now in this little picture, it looks like it's targeting a bunch of things, but really the other things that it's targeting is mostly in the liver. Okay. The book says this, and I haven't been able to verify it with other sources, that glucagon has a general effect across the body, but I've not found any other supporting evidence of that. I think it's all limited to the liver. And what it tells the liver to do is to release glucose back into the bloodstream to get blood glucose levels up. Liver stores glucose in the form of a starch-like molecule called glucagon. Okay? It also stimulates in the liver something called gluconeogenesis. And why they have pictures of the ER here to represent that, I don't know, and I don't care. But in the liver, taking the remnants of glucose metabolism, the breakdown products of glucose, the liver can actually rebuild glucose out of it. It's not a very energy efficient thing to do, but it does get blood glucose levels up, and it moves glucose from the liver back out to the tissues that need it. So it's something that glucagon can stimulate. So whether the liver is releasing glucose into the bloodstream from glucagon or building new glucose from the breakdown products of previous use, it brings the blood glucose levels up, and we get back to the homeostatic set point. If blood sugar levels are high, 160 milligrams per deciliter as an example, then the beta cells that will release insulin, and that actually does target everything in your body. It tells every single cell in your body to take up the glucose and use it. Okay. It further stimulates the liver to take glucose and build glycogen to store up for when it needs to release it in the future. And it inhibits the use of other molecules to make glucose. So it sort of counteracts the glucagon effects down here. Okay. What's not mentioned here, because it's not usually associated with blood glucose levels, but it's true of insulin, it also stimulates the uptake of amino acids and lipids into cells. Okay. Now, when you eat, blood glucose levels go up because you just had a big meal, stimulates insulin release, and insulin tells your cells to take up glucose. But if you just ate, you probably also have an increase in amino acid and lipid levels in your blood too, because probably all three of them are in the food that you're eating, unless you live strictly on pixie sticks. And so insulin, since it's already telling cells to take up glucose, is going to go ahead and tell cells to take up those other nutrients because it's efficient use of that signal. But the book is just concentrating on the glucose part because that's the real homeostatic mechanism that we're talking about here. So that's sort of a figure 8. You eat food, levels go up, insulin's released, insulin tells everything in your body to use that glucose. And then the blood levels go back down. They continue to drop off, and eventually glucagon's released to the liver to get those levels back up. Okay. And it just sort of keeps going back and forth in the figure 8. Now you're helping this because what are you doing to raise glucose levels? Eating. And what are you not doing when glucose levels drop? Eating, which we call fasting. Okay. So when you eat, you're pushing yourself into the insulin side of things, and when you fast, you push yourself into glucagon side of things. Okay. that's a quick little explanation of pancreas function, which I hope covers everything. And I did that because I wanted to not go here, go here, and talk a little bit about growth hormone. Okay. Now, this first picture is not out of the book, it's off of Wikipedia, but I like it as a little summary of what growth hormone does. Growth hormone is released from the pituitary. It's released from the pituitary because the hypothalamus tells it to. Okay? There's a releasing hormone that causes growth hormone release. Growth hormone has growth effects. There are some direct effects that it has on some tissues, and then there are indirect effects that it has through an intermediate called IGF-1. Okay. Now, I'm going to try to explain this kind of quickly because I think it's important for you to hear this. It's hard to really get this out of what the book says, but IGF-1 stands for insulin-like growth factor 1. Now, the one part doesn't really matter. There's two versions of this molecule, but we just care about this one thing. What's important is that it's called insulin-like growth factor. And I'm telling you about this now because we just learned about insulin. This hormone, this protein, came into existence because sometime way back in the past, an organism was reproducing and it copied DNA to make a new cell, and the insulin gene got duplicated. And that organism needed insulin to do what insulin's supposed to do, but it had an excellent extra gene which wasn't necessary, so it mutated. Okay? And it mutated into a variation that's not insulin, but it's very close to insulin. So it's called insulin-like growth factor. What insulin-like growth factor does is it causes tissue growth. In the picture here, it kind of is emphasizing bone growth because that's what we associate it with most. So remember in A&P1, when you're learning about the skeleton and bones, there was that cartilage growth plate. Remember that? What stimulates that cartilage to grow so that a toddler grows to full height is insulin-like growth factor. It causes the cartilage cells to divide and the tissue to eventually calcify. It does that because growth hormone tells the liver to make IGF-1. Okay. You are releasing growth hormone at night. Okay, it's tied into the circadian rhythms when you release it. And so overnight, you release growth hormone. It tells the liver to make IGF-1, and your bones grow. Now, none of you are doing that because you're all full grown. But when you were a child, that was happening. Growth hormone was released, and it caused your bones to grow. But your growth plates have fully calcified, and your bones are as long as they're going to get. Most likely. I don't think anybody in here is not done growing. By the time you're 15 to 18, you finish things up. But if you're in here and you're 17, maybe you're still growing. I doubt anybody's still 17 in AP2. But so probably everybody's past growing. But during adolescence, that's what's going on, okay, and childhood. Now, in the book, they have this figure here that gets into what growth hormone does in a little bit more detail. And I want to take this apart really quickly in 5 minutes to help you understand what's going on. Okay. So first off, the hypothalamus releases a hormone called GHRH, which stands for growth hormone releasing hormone, which is released into the bloodstream and travels down to the anterior pituitary, where it tells the anterior pituitary to release growth hormone. Okay. Growth hormone then goes to the liver and causes IGF-1 release. It also tells the liver to break down glycogen and release that into the bloodstream. Okay. Growth hormone can actually, and I didn't realize this until I had to look it up when I started using this book, growth hormone will actually directly cause some growth effects. But IGF-1 does that too. Now, the reason I didn't know this until I got to this book is I used to work in a laboratory that studied growth hormone. And we constantly talked about IGF-1 being the growth effect mediator. Okay? Growth hormone didn't cause growth, IGF did. Growth hormone caused IGF, which caused growth. But since I worked in that laboratory, apparently studies have come out to show that growth hormone does in fact have some direct effects. The studies that I could find that supported that all came from the 2000s, which was definitely after I was working in that lab. So it's new since I was working there. And really the best explanations have only come out in the past 5 or 7 years. So in the book, they kind of downplay the IGF role, but it does play that. And also those studies showed that growth hormone can cause growth and IGF can cause growth, but really the best growth comes when the two are together, which is usually going to happen anyways in normal situations. Growth hormone also causes adipose tissue to break down stored fats, releasing more energy-rich molecules. Now, this diabetogenic effect, okay, it's a weird word, but really what it means is it raises blood glucose levels. Okay. What did we recently learn also raises blood glucose levels? Nope, but you're close. Glucagon. Insulin lowers blood glucose levels. It responds to high blood glucose levels. Now, when you're diabetic, it's because you don't have insulin to lower blood glucose levels. Okay? So diabetogenic effect means raising blood glucose levels like you would see in diabetes. But it's really mimicking glucagon's effects. Okay? And growth hormone does that, and growth hormone also stimulates fat metabolism because growth processes need energy to take place. Okay? And IGF, insulin-like growth factor, causes some of that growth. But also, IGF is like insulin, which means that it stimulates insulin receptors, which causes a drop in blood glucose levels. Okay? So while IGF is causing growth, growth hormone has to counteract the insulin-like effects by increasing blood glucose levels and increasing fat metabolism. So there's energy for growth. Okay? Now, that helps with growth as when you're a kid. When you're an adult, this stuff is kind of still happening, except the growth is kind of stopped. But growth hormone's still released, and it still has its effects. IGF is released, but it doesn't have effects on body growth. It has more effects on, brain function and that sort of thing. And, helping neurons change and adapt to learning and that sort of thing. But when did I say growth hormone is released? At night. What are you not doing at night? Eating. Okay. So growth hormone, while it's released at night to stimulate growth overnight, which is what happens when you get taller at night, it also helps to supplement glucagon's effects while you're definitely fasting overnight. Okay. So as an adult, growth hormone's more about helping keep blood glucose levels under control. Because you're done growing, but it can still do the diabetogenic thing and help glucagon overnight when you're not taking any new food. Okay, so that is a quick explanation of things. I hope it hits on the big points so you understand a little bit. There are a couple of other things in this presentation. I need to find a good picture. The one that's in here right now is copyrighted, but this is showing the calcium homeostasis story with thyroid, parafollicular cells in the thyroid, and the parathyroid gland. And then this table is at the beginning of the chapter, and it's just listing differences between endocrine and nervous system function. So just to point those out. I'll try to get some presentations posted with that stuff, explain a little bit more in depth. But otherwise, have a nice week.